All right, so our next speaker is Rob Holt, who received his PhD in pharmacology from the University of Alberta in Canada in 1998. After a brief postdoctoral fellowship in molecular evolution at the State University of New York, Dr. Holt joined the company Celera Genomics, where he served on Craig Ventnor's team as the scientific operations manager for the initial sequencing of the human genome. Since 2003, Dr. Holt has been a scientist at Canada's Michael Smith Genome Sciences Centre, located in the British Columbia Cancer Agency, where he also co-directs the immunotherapy program. His current basic and clinical research directions are focused on synthetic immunology and immune interventions in cancer, and also healthcare implementation research. And welcome.
Thanks, Steve. And thanks to Jamie and Felix and the organizers for the invitation to speak today. This has been a great trip. I had a few days in Tuscany before coming to Genoa, which was very nice. So here we are. Let me just check the time. So we're starting a few minutes early, so I've got lots of luxurious time to go into excruciating detail, all of these slides. Okay, so it's really amazing actually how this field has grown over the past few years, continues to grow. It's very exciting. So our own work has taken recent years much more of a clinical direction. So we are focused very much on translational immuno-oncology work, and I'll take you through a little bit of the background and a couple vignettes around some current challenges and observations through case studies. Okay, so here's a tumor section by IHC, and it's from high-grade serous ovarian cancer, and it's quite typical for this disease and for many other solid tumor types, particularly epithelial cancers. So there's often an immune infiltrate, right? So there's infiltrating T cells and B cell subsets stained here. So the CD8's in brown, right? Green is a few CD4 cells and some pink B cells. So if you stratify patients based on the density of those immune infiltrates, there's quite a striking survival advantage to having a dense T cell infiltrate. And this effect is dominated by the CD8 T cells, recognizing class 1 antigens. So that's true in high-grade serous ovarian cancer and essentially any other solid tumor that's been looked at in this way for the past couple of decades. It's quite well established. So the question for quite a while was, what were those infiltrating T cells recognizing? What were the class 1 antigens? And it's become reasonably clear recently that they're neoantigens. So they are peptide epitopes that contain amino acid variants derived from tumor mutations, for the most part. There's other types of antigens, but these tend to be the predominant type of antigen tumor infiltrating T cells, the CD8s are recognizing. So here's one view that we had a few years ago. So it was the first sort of holistic view. So we were part of the Cancer Genome Atlas study through NIH. So we had an early look at the data. This is 7 different tumor types. And so what we did was took CD8A gene expression from, it's about 500 patients, plotted that and ordered by decreasing CD8A expression as a surrogate marker for CD8 TIL in the tumor environment. Okay? And then for the same patients below, plotted the neoantigen frequency, basically. So that's where we determined each patient's HLA type extracted from the RNA-seq data, predicted which of the mutations would be presented by their autologous HLAs, counted them up and plotted them next to their TIL density. So there's not a perfect correlation, but quite a striking relationship between TIL and neoantigen load. So this has been replicated more recently in the complete TCGA data set and in lots of other cohorts. Interestingly, another line of evidence that neoantigens are key comes from checkpoint blockade inhibitors and the observation that they work better in patients with a higher mutational load. So here's the first study that showed that for ipilimumab, CTLA4 blocker, and they just arbitrarily stratified in their cohort based on, sorry, you can't really read that, that says 100 mutations or more, or less than 100 mutations in the sequenced tumors from their patients. So there's quite a striking survival advantage, or sorry, a response to the drug in this case, if there's a higher mutational load. And again, this has been shown in many other studies and for other checkpoint blockade inhibitors, including the PD-1 blockers. And these are the drugs, of course, that work by disinhibiting T cells, exhausted T cells, non-responsive T cells in the tumor microenvironment. Okay, so we tried to put these things together more recently. So we did quite a deep study with Saurabh Shah and Brad Nelson and others at our cancer center, where we did deep sequencing of tumors sampled over time and space in 38 high-grade serous ovarian cancer patients. Okay, and even at the different sites, we took sub-regional sampling. So we had quite a large spectrum of tumor samples to compare. So from the mutation profiles, the tumor lineages were reconstructed to show the clonal evolution within each patient. And this is depicted in the following figures by these donuts here that just show the composition of each tumor clone, or sorry, the composition of each tumor sample in terms of what clones make up or are represented in that sample. So if we look at the right side of the slide here, so this is from one patient. So the ovary sample 1 and ovary sample 2 were similar but had slightly different clonal composition. And it was quite different from the clonal composition in 2 omentum subsamples. So we did TCR-seq, or I guess what we call AIRR-seq now, for all the patients where we had access to the materials, which turned out to be only 13 of them, and where we had the TCR-seq data and the genome sequencing data from at least 3 samples from each patient. Okay, so if we look at that, sorry, this is a bit complicated, but what's shown clonal composition rate, and the bands here, the black bands, are the immune repertoire, the T cell repertoire in each of those tumor samples. And here, by these interconnecting bands, it shows what's shared versus what's private, or here what's shared versus what's private in terms of T cells seen in that patient. This is from beta chain sequencing. Okay, so in this example, we see there's some clones here that are shared only between the 2 omentum samples, but not between the ovary samples, and some in the ovary samples shared with each other, but not with the omentum samples. Okay, so there is here a relationship between TIL composition and clonal composition of the tumor. So I apologize for the color key here. Okay, so these colors here refer to publicity. So this orange-ish color means these clones are seen in just 2 samples. Okay, this orange, which looks quite similar to that orange, unfortunately, is referring to the clonal structure. Okay, so if we do that and look statistically at all of the data, that there is a relationship between TIL content and tumor clonal content in 7 of the 13 patients. And that's predominantly in the patients with the highest TIL density. So looking at another patient, it's quite different. So this patient has clonally pure tumors at the different sites and quite a diverse TIL repertoire in each of them, and quite a lot of overlap in the T cell clonotypes present at the different tumor sites. So that's in contrast to the first patient, where we could actually map out a relationship between the T cells and the tumor mutations. Okay, so what's interesting in this case is that one of the tumor subclones here in the right ovary had quite a striking loss of one of the HLA haplotypes, shown here in this copy number plot. So it's another piece of evidence, I suppose, that there is T cell pressure against neoantigens that's perhaps driving the evolution of these different tumor samples as they evolve. So that's great. So these types of observational studies can reveal these types of trends, right? So they can highlight the fact that neoantigens are important, that we should be looking at neoantigens. You can also profile T cell phenotypes, exhaustion markers, other T cell subsets, and how they track in the microenvironment. So not really showing any of that today, but the fact is that that doesn't tell us which specific neoantigens are recognized in by which specific T cells. Okay, so that becomes important if we want to actually do anything of therapeutic relevance with this information. So we can't take a T cell therapy to the clinic based on a probability that the T cell recognizes something in the tumor environment. I mean, that has been done in previous decades, but that doesn't pass the bar currently. So when we've set up a system for manufacturing, for GMP manufacturing of genetically engineered T cells, and taking those into phase 1/2 clinical trials. And we have to decide what we're going to spend $1 million on to make a lentivirus using GMP manufacturing, and what we're going to spend several $1 million on for the clinical costs of running the actual trial. So that can't be probabilistic. We actually have to know what T cell is recognizing which antigen. Okay? So we do a lot of computational work, but unfortunately the computational methods don't yet give us that precise mapping. So we have to resort to conventional approaches. So I'll show you a couple ways that we do that here. So here's a case study in high-grade serous ovarian cancer. So this is a difficult disease, right? So the median survival is only about 18 months. It's usually diagnosed late. And the therapy right now is old. It's based mostly on taxanes. It can be very effective. So in this patient, we're tracking her response using CA-125. So that's MUC16. It's a surrogate marker for tumor load, tumor burden, tracked quite easily in peripheral blood. So at her initial diagnosis, she was treated with carboplatin and paclitaxel and had quite a complete remission that lasted for about 6 months. And then there was a recurrence of the cancer, which is quite typical, right? So she received another treatment regimen here, carboplatin and gemcitabine, and it was quite effective, but only for a short period of time. So there was remission for a few months, and then a final recurrence, and unfortunately she died shortly thereafter. So we looked at this retrospectively, and we gathered through the course of her treatment, blood draws, and vast numbers of cells from the ascites. So that contains both tumor cells and immune cells. Okay, so we did exome sequencing, about 100x coverage, found the mutations, about 40 mutations, non-synonymous point mutations, amino acid changes, and those are quite stable across the 3 time points, the primary disease in each of the 2 recurrences. They changed in frequency, but there were actually none of those mutations that were gained or lost during that evolution. Okay, so we had her bulk T cells from the 3 time points, so they were expanded, and we tested predicted neoantigens from spanning each of those mutations. And we found one fairly strong hit to this gene, HSDL1, which is a very obscure, uninteresting gene, hydroxysteroid-like dehydrogenase 1. But for her, it made a nice class 1 antigen, and there was a T cell response very selectively against that one mutation. So this is representative of what's seen in essentially all of these types of neoantigen explorations. If you look retrospectively across tumor mutations in any given patient to see which neoantigens are recognized by a T cell, you map it out like this, there's always 1 or 2 that are immunodominant. There could be 10, 100, close to 1,000 different mutations in the patient's tumor, but for reasons that aren't understood, one of them tends to dominate. So this is a clear example of this here. So it gets even more interesting here. So if we look at these columns, primary disease, recurrence 1 and recurrence 2, we see that the HSDL1 mutation frequency is quite low. It's detectable, but it's quite a minor allele in primary disease. And then it becomes more prominent, up to about 50% and slightly higher in the recurrences. So that tumor clone is expanding. And then the bulk T cells expanded out of the ascites recognize, sorry, that's at the bottom here. The bulk T cells are recognizing only the second recurrence. Okay, so her T cells, expand them, they don't recognize the primary disease, and they don't recognize the second recurrence. But when we take those T cells and we clone out the individual T cell reactive to HSDL1 and test it back against those samples, it does recognize the third recurrence, or the second recurrence, the 3rd time point, where we know the mutation is still expressed. So somehow that T cell was lost from her repertoire, and this is just another way to look at that. Primary disease here, very low frequency mutation. We can't detect that T cell clone either by ELISpot or by quantitative PCR that clone it to type specific primers. First recurrence, increase in mutation frequency. That clone appears to be reactive, and it doesn't become hugely prevalent in the population, but it expands. Mutation persists as that cancer evolves, but the T cell is simply lost. So we have no explanation for that. So in our previous study, I showed how you can have HLA loss variants that can arise from time to time. Here, that's not the problem. The mutation is still recognizable if we isolate the clone, but naturally in her repertoire, the T cell has disappeared. So these are things we don't have answers for. So why was immunity detected only against that one immunodominant antigen? Why that one of all of that list of more interesting genes? Why did it fail? Why was that T cell lost? And how could we exploit this in future? So if we were doing this prospectively, not retrospectively, you can imagine perhaps isolating the T cell clone at the time when it predominates at the first recurrence and having it on hand to use as adoptive cell therapy at the time of the second recurrence. So that we would have predicted could have an effect in this patient, or even beforehand. What if you give it here in a preventative type of approach? So we didn't have that opportunity. There's another problem, however, and this would be extremely personalized therapy, right? So the presenting allele, we map this out using a panel of LCL lines, is quite rare. So it's HLA-C 1403, so that's less than 1% of the population. Okay, but so if all high-grade serous patients had an HSCL1 mutation in this hotspot, we could only treat 1% of them with that TCR, because they would only have the restricting allele. Okay, but the mutation is completely personalized. So in all of COSMIC and all of TCGA, there's no other HSDL1 somatic mutation. So this is truly an individual therapy. So that's not particularly feasible. So the view is actually quite stark if you want to look at it as anything other than personalized therapy. So this is all of the TCGA data from 27 tumor sites, 5,000 subjects. And this is the number of subjects that have each of any given mutation. So this is restricted to those that are recurrent. So they're seen in at least 2 patients of the 5,000. And any patient can have between 10 and several thousand mutations. So if you were actually to stretch this out and include the singletons, it would go across to the architecture building where we had lunch the other day. So there's actually only a handful of mutations with meaningful recurrence, so hotspot mutations in all of cancer. So this is really the take-home message from the Cancer Genome Atlas project. So we thought, let's do something more sensible and try and focus on this end of the spectrum. So we're looking at the KRAS codon 12 mutations. So this is the most predominant hotspot mutation in, well, all of cancer. So it's particularly prevalent in pancreatic ductal adenocarcinoma. Okay, so more than 95% of pancreatic cancer patients will have a codon 12 mutation, either G12D or G12V, sometimes G12C. And this too is a very difficult disease. Right? So this is the incidence, new cases, and the number of deaths spanning back to 1975. So it's remarkably unchanging. Okay. The 5-year survival has gone from 3% to 7% at the end of the plot year 2006, and it's currently about 9%. So it's probably the most deadly cancer. And yet it has a nice target that's just sitting there that has been completely untargetable. So these, so RAS was identified as an oncogene quite a while ago in 1983. And for the past 35 years, there's been intensive efforts in the pharmaceutical industry to develop a RAS inhibitor. These have not been effective. Nothing has made it to the clinic that has shown any efficacy in terms of a small molecule drug inhibiting KRAS. For lots of reasons, it's difficult, right? So RAS is a GDP-GTP exchange factor. So it's a signal transduction molecule right in the epidermal growth factor receptor pathway. Links that to the mitogen activated protein kinase pathway. So it's small and seems insignificant, but it has a key role, and the codon 12 mutations lock it into the activated GTP bound state. So it produces tonic signaling. So it's constitutively activated signaling, so continuous growth signaling, and that's the oncogenic signal that leads to cancer in these cases. So there's been lots of attempts. So for example, RAS is farnesylated. It's implanted inserted into the membrane. So farnesyl transferase inhibitors have been tried to prevent that. They haven't worked. Drugs to actually dislodge GTP from the site haven't worked because GTP, once it's bound to the mutated codon 12 position, is just undislodgeable. And other targets where small molecule drugs have been tried to be inserted to just change the general conformation to get it to give up GTP. None of this has worked. So we're hoping that this could actually be a reasonable immunological target, even if it has not been an approachable pharmaceutical target. So here, again, we need to actually find T cell receptors that will recognize neoantigenic peptides containing the codon 12 altered residues presented by hopefully a reasonably abundant HLA class 1 allele at the cell surface. Okay, so this is the process we use to isolate neoantigen reactive T cells. So 9 easy steps. So first of all, we predict epitopes, and then we use HLA elution and peptide elution from cell lines expressing the mutation with the appropriate HLA type to verify that the epitope is actually processed and presented by that HLA. Okay. Then from patient PBMC, we do what we call as a mini line screen. So we dispense about 2,000 T cells per well across multiple plates, expand them using high dose IL-2, CD3 antibody, so non-specific expansion, let them rest. Then we screen back against the verified antigen using ELISpot. Then if we see a positive hit in one of these wells, we go back to the master plate. We take that well, we re-expand it, and then we do single cell sorting. So we're looking for activation against the peptide target indicated by CD137 upregulation. So we flow sort based on that activation marker back into individual wells within 1 cell per well, expand those, take several weeks, then re-verify the specificity of the neoantigen recognition, again by ELISpot and by tetramer at this stage and by cytolytic assays. Sequence then the alpha beta TCR from that clone, make lentivirus and reconstitute the clone in healthy donor lymphocytes of the same HLA background, and then verify the function of that recombinant TCR. So all of that takes a while, months. Okay, so for KRAS, we went through this process. So we picked some codon 12 mutation candidate neoepitopes, verified them. One was particularly dominant, so we screened against that one, and we came out with a panel of 3 different TCRs here. So from this one patient, 1 was reactive to a G12V mutation and 2 to a G12D version of the peptide. So here, this is just showing the G12D reactivity and clone 2, right? So here's tetramer staining against wild type peptide against, T cells recognize the tetramer with the wild type peptide recognizing this T cell clone, no signal, G12V, no tetramer reactivity, and here's the G12D tetramer staining showing quite strong positivity, and by ELISpot, PANC1 cells naturally process and present the G12D peptide, the AO201 positive, which is our HLA allele of interest here, versus HeLa cells, KRAS wild type, and AO201 positive. So that is looking good for the T cell clones at this point. Then we went through the remaining steps and reconstituted those alpha beta TCRs, and we're able to recapitulate selective binding quite well. So here's clone 1, right? So it's G12V specific with no reactivity against G12D or against wild-type peptide. And here's the G2 G12D specific KRAS clones showing selectivity for that mutant peptide, both by ELISpot and by cytolytic activity. Okay, so now we have recombinant TCRs that are looking interesting. But it gets a bit bizarre at this point. So here's the grid I just showed you. So first of all, it's peculiar that we can get selective mutation reactivity against two different mutations from the same patient. So you would tend to think that a patient that incurs a RAS mutation would incur one mutation and not more than one. So we went back and we reviewed all the sequence data carefully, and there really are two distinct T-cell clones in this patient, and both mutations. You would never find this in TCGA, for example, because there's actually no way to report more than one minor allele. So it's perhaps not seen as something that's prominent because it's overlooked in many of the large data sets. Okay, so that's a bit of a tangent, okay? But what's interesting here is we've got two of the same beta chain. Where are we here? So we've got TCR beta 19 for two of the clones, and they have different alpha chains, right? So that seems straightforward. We could have one T cell with two alpha chains. About 20% of T cells have two alpha chains. And that could be conferring selectivity, the alpha chain, for B or D mutations. That wasn't too peculiar, but then if you look further, we've got two alpha chains here that are the same, but different beta chains. And both of those recognize the G12D mutation. So we have no explanation for that. Again, we went back carefully, looked to see if we had maybe two clones in the single cell sorting, contaminating each other, but no, the sequencing data is quite clean. So there isn't any known biological explanation for having one alpha chain and two different beta chains in a T cell or across two different T cells. Seems peculiar that they would make and select the same alpha chain completely independently, but I guess that's a formal possibility. Okay, but if you look at this grid, so we've got two different alpha chains and two different beta chains. My battery's dead. Is that a hint? Okay, the pit crew is here. So we flip this inside out, okay? And you'll see that in a moment. But we have two alpha chains and two beta chains and three TCRs. So that leaves one combination of an alpha and a beta chain unaccounted for that we didn't find in nature, but we would predict may have some anti-KRAS activity. So that would be alpha 2701 from the TCR1 paired with beta chain 04 from TCR3. So if you look at that combination, it doesn't appear in anything we isolated or sequenced, but perhaps would. Thank you. Okay, one beta chain, two different alpha chains, two alpha chains, one alpha chain, two different beta chains. We flip that around. We've got these combinations accounted for, but we didn't find this one, but we would predict that alpha 27 and beta 4 maybe does something. So we reconstituted that TCR by lentiviral transduction into healthy donor T cells, and in fact it does, right? So this is the previous plot of reactivity by ELISpot for KTCR1 and 2 and 3, And this is that novel or unnatural synthetic combination. So it has quite strikingly stronger binding or activity in ELISpot for both G12D and G12V, which is a big surprise. So Craig, who's the postdoc's been working on this, he calls this mayhem. He says it's like cats and dogs living together. It makes no sense. But it's potentially a very valuable therapeutic, right? Because you would like to be able to target both mutant alleles when they occur with one infusion product, right? With one lentiviral prep. So it needs a lot more characterization, right? We just got this final result last week while I was on the way here. And it's striking because you wouldn't really, we had no idea what to expect, right? Maybe putting those two chains together would abolish binding altogether. Maybe the alpha chain would dominate, it'd be D specific. Maybe the beta chain would dominate, it'd be V specific. So it was really quite interesting that we got probably the best possible result, that it's stronger for both of them. Okay, so this can't go straight into the clinic. These, we actually have a path to a phase 1 clinical trial that's being initiated now for pancreatic ductal adenocarcinoma in late stage patients. These, because it's truly synthetic and not natural, we have no way to predict what the off-target binding might also be, which can be quite problematic with affinity enhanced or otherwise modified TCRs. So that's work that will be ongoing, but it's, I thought, quite an interesting result. So just to wrap up, summary analysis of tumor-associated T-cell repertoires is very useful, can show interesting trends, can just indicate where you should focus your efforts for functional studies. Mutation-specific T-cells can be recovered from patients using conventional methods. Hopefully, the computational methods will improve to the point where we can get very strong candidate TCRs computationally. There's some hints that that will be coming, and I would really look forward to that because it is quite a tortured procedure to do this with conventional lab-based approaches. And even novel specificities can be obtained by creative TCR reconstitution, we call them. So I'd just like to thank our various funders and members in the lab. This is where our lab is at the BC Cancer Agency in Vancouver. If you ever want to come and visit, that would be fantastic. Craig Rivet here did the bulk of the work with help from Scott Brown and Eric Young on the KRAS project. Scott Brown is a computational biologist, did all of the heavy lifting for the TCGA analysis. And Govinda Sharma is here at the meeting. And he has a poster at lunch I'd encourage you to see. So we published our first T-cell repertoire analysis paper 10 years ago. Felix just reminded me of. And actually since then, we've been working on how in the heck can you link T-cell receptors to T-cell epitopes using high throughput genomics methodologies. So Govinda's actually reduced something to practice now after quite a few years of work called TOPE-seq. So have a look at that if you're able to at the poster session. Thank you very much.
We've got a couple minutes for questions. Maybe let me start off with one question. With going back to the TCR repertoire profiling at the different tissue sites, were you also looking at blood? I mean, in the cases where you could track them among multiple tissues, could you also see transit into the blood?
No, we actually didn't profile blood in that study. It was, so sorry, Steve was just asking if you didn't hear that, whether any of those T cells were detectable in blood and what that looked like from the regional sampling of high-grade serous ovarian cancer. And sorry, we didn't profile that. When we do look at blood, so these are very difficult to detect. without really deep sequencing in blood because the diversity is so great. And when we detect them by cloning out of blood, it requires enormous expansion, right? Before we can select T cell clones, the whole population needs to expand in many log before we can isolate them.
So I was, to that point, what about focusing on the PD-1 subset? So sorting first PD-1 positive T cells, which I think have been shown to be enriched. And then also regarding the IL-2 expansion, do you notice any narrowing of the repertoire after the non-specific expansions? If you compare the diversity of your starting pool and your post-expansion pool, does it narrow?
Okay, the first question, yeah, in other studies we are looking at PD-1, which is an activation marker, you're right, also before it becomes an exhaustion marker. But we find CD137, is what we're doing here to pick out that subset of activated T cells, is actually more effective, just empirically. It gives you a stronger signal, comes up faster, so it works well for that purpose. And sorry, what was the second question? Oh, yeah, we actually haven't looked at that. You're right, it probably is restricted. We're probably recovering, after these expansions, just a subset of the repertoire, but we haven't characterized that.
Yes, so in this algorithm here for making a T cell line that you could put into someone and go after their neoantigens producing tumor cells, I'd like to know, you're saying, right, the penultimate there is reconstitute TCR in healthy donor T cells.
Yeah.
So when you say healthy donor, do you mean somebody else who is not the cancer patient? And if that's the case, then, or do you mean healthy donor T cells, meaning from the cancer patient? Because otherwise you've got a little bit of a, transplantation
Yes,
issue.
sorry, that's not what I meant. So this is for the purpose of characterizing that TCR by ELISpot and tetramer staining.
Okay,
right?
so
it's not the clinical approach.
So you would take the person's own T cells then?
It's an autologous cell therapy, yes.
Okay.
So if we have the lentivirus, we can transduce their cells like we're doing currently for CAR T therapy.
I just have a question because I think there are like different opinions in the community. So in your opinion, if you have in one person the same CD3 region but different V genes, so because you have worked a lot on this, do you think it binds the same antigen? You have the same CD3 but different V genes?
Yeah, that's a great question. And just let me clarify before I answer that. I refer to these clones by their their alpha and beta chains, okay? But they are the same clonotype. They have the same CDR3 sequence. So we're talking about T cell clones here. But your question is a bit different. Is if we happen to have the same CDR3 but different flanking V and J genes, would the specificity be the same? So we've taken a very conservative approach in all of our repertoire profiling, and we consider those different clonotypes with different binding. Well, we don't know what the binding specificity is, but potentially they are of different binding specificities, so we consider them that way. Another point is that you can actually have mutations through TCR mutagenesis studies outside of CDR3 and even outside of CDR1 and 2 that change binding specificity. Individual point mutations. So David Kranz's group has shown this quite convincingly. So I don't know, but I'm being cautious and saying probably, at least that's what we consider the case to be.
Okay, thank you.
All right, let's thank our speaker again.
Thanks.